So I think I will begin. Sydney, are we live?
We are live.
Okay, great, thank you. Welcome everyone to the university lecture for fall 2020. This year's lecture could not be more timely. Most of you on this call are asked every day that you plan to visit campus to check off a list of symptoms that includes a question about whether you have a persistent cough. At this point, I believe we are all highly aware of how COVID-19 can impair respiratory function, sometimes fatally. That question is a red flag. Our university lecturer of 2020, Dr. Joseph J. Mizgerd, will speak about respiratory infections. As you will see from the online program, his lecture is entitled From Cough to COVID, How Respiratory Infections Produce Problems and How Our Bodies Fight Back. Dr. Mizgerd is professor of medicine, microbiology, and biochemistry at the School of Medicine and director of the Boston University Pulmonary Center. The short description of his work is that he studies with his team the immunology in the lung and its influence on acute lower respiratory tract infections. He earned his undergraduate degree at Amherst College and his doctorate of science at the Harvard School of Public Health, where he was first a postdoctoral fellow and then a member of the faculty until 2008, when he joined Boston University. The university lecture dates to 1950. It was conceived as a way to honor Boston University's most distinguished scholars and researchers, and perhaps more importantly, to bring their work to the attention of the university community and to the broader public. That is, to inform the public from the basis of sound research and carefully developed scientific expertise, as is the case tonight. This is what Dr. Mizgerd brings to us tonight, during a time when we are all of necessity focused on the well-being of our community, our nation, and the world. Dr. Mizgerd, I give you the platform. Thank you.
Thank you very much, Dr. Brown. I'm really pleased to be with you all today. I'll share my screen. And does that look right? Is that the cover screen for everybody? I'll take that as a yes. So I'm really pleased to be here today. Thank you all for inviting me and giving me this opportunity. This is a special time for all of us because of COVID-19, and it's unfortunate that I'm having to give this remotely, but I'm still so pleased that I get this opportunity to share some of my science with this large group, even if remotely. So thank you to President Brown and to Provost Morrison for this opportunity. And thank you to all of you for being here today. On the left here is a very beautiful poster that was put together by the BU team that shows a set of lungs inside of the chest cavity, and it looks like they're glowing embers. And I think that's very apropos. That really is what is happening in the lungs. They're inflamed. And let me see, I'm trying to move that. There we go. And so that's a beautiful, fanciful description of what the lungs look like during pneumonia in our imaginations. On the right-hand side here, though, this is actually a picture of what lungs do look like during pneumonia. This is a real set of lungs from a patient who unfortunately died of pneumonia. And each of these different colors is a different type of cell, which we've highlighted in aqua or green or purple or red or orange in order to identify what kind of cell it is in this lung. And they're all different immune cells, and these immune cells are working together to try to fight the infection that's occurring in this lung. And that's what we're going to focus on today. But I think we need to start by emphasizing the infection that brings us all together but apart today, the reason we're doing this remotely, which is COVID-19. COVID-19 is having a tremendous impact, as Bob mentioned, and all of us recognize that, and I don't need to go into detail about that, but I think I need to start with that just to set a perspective. it's hard to understand or hard to know how to best communicate the impact of pneumonia, of COVID-19. And one way that we can think about this is deaths, and this is death rate in the United States. And you can see the death rate in the United States beginning in 1900 up through 2020. It's been, for the most part, this is all-cause deaths, nicely decreasing, march of progress for 120 years with just two exceptions. And those two exceptions really are the influenza pandemic of 1918 and now today in 2020. And I want to emphasize that this is all-cause deaths. The number of deaths is increasing dramatically, no matter what you want to call responsible for those deaths. There's too many deaths in the US right now. And this is a vast underestimate at present. These data only calculate the deaths that occurred through August of this year. A lot have occurred since, and as we all know, the numbers are increasing. The numbers of COVID cases per day have doubled since November 1st in the United States. And after the cases increase, the hospitalizations increased, and then the deaths increase. It's surely going to happen. This number is going to be much higher. We don't know how high it'll go, but this is a generation-defining experience we're all in the middle of. Well, what is causing these deaths? These are the most common causes of deaths over the last 5 to 6 years. And you can see most of them are pretty much similar across all the 5-6 years, including here in 2020, with this one vast exception, the bright pink line of COVID-19. COVID-19 began in the US in early part of the year. Deaths increased dramatically until in a couple weeks in April, it was the #1 cause of death throughout the entire United States. It dropped again over the summer and it's rising again now. So we don't know where it will end up, but it's the third most common cause of death in the United States, and it's rising. And deaths, unfortunately, are really only the tip of the iceberg. They dramatically underestimate the impact of this infection. Only a very small number of people die due to COVID-19. For every one person who dies, 100 survive, and those survivors suffer terribly. Many of them in the ICUs will have acute respiratory distress syndrome, meaning they need to be on ventilators. They'll suffer sepsis, meaning they have organ failure, such as kidney failure. These can trigger acute events like myocardial infarctions, atrial fibrillation, and strokes, all of which can have devastating consequences for individuals. If they survive these situations, even then, there can be long-term consequences, what's often called long COVID now, or COVID long haulers. There's loss of pulmonary function, loss of cognitive function. There's emotional and mood and mental health problems. And all of this can persist for long periods of time. It's only been months now, but we know from other types of critical care illnesses that these can be very long-term diseases that resulted from the acute effects of the infection. In addition, even for those people who aren't suffering from COVID-19 itself, there's tremendous financial loss and tremendous costs of social isolation that we're all experiencing. So this, I think, I don't have to argue with anybody in the room about this. COVID-19 is having a profound and terrible impact on our community. So what's so different about COVID-19? We've all experienced respiratory infections before. Why is COVID-19 so different? There's really two things that are very different. First is it's really exceptionally good at spreading. It spreads tremendously well. And second, in addition to causing a lot of infections, those infections often cause a lot of disease. And that's highlighted, the principles underlying this is highlighted in this recent perspective from Lancet Infectious Diseases. It's a highly transmissible virus, SARS-CoV-2, the virus that causes COVID-19. It has a long incubation period before you feel any symptoms if you get infected. So that means people are walking around and infecting each other. The interval between when the symptoms start and your maximum infectivity is actually 0 or even negative. The peak infectivity often occurs before and in the very first days of symptoms. And so people very often don't know they're infecting others. And often, because so many people have a very mild illness, they don't even know that they should be sequestering themselves. And so again, all of these together lead to an exceptional spread of this virus. And then it's an unusually disease-causing virus. Not a world beater. Some infections like SARS-CoV, which was the initial coronavirus that caused SARS in 2003, that coronavirus was really very severe. If you got that virus, you were very likely to end up hospitalized. This has a much lower rate of hospitalization than SARS-CoV-2, but it still has a very high 20% hospitalization is a very high rate compared to even things like these pandemic influenzas, a much higher rate of hospitalization. So it causes a lot of disease. And what kind of disease does it cause? It causes pneumonia. And pneumonia was a big, big problem long before COVID came along. These are all data, all the data on this slide are pre-COVID statistics. This is global burden of disease measured using disability adjusted life years lost, which is the favorite metric of the World Health Organization. And they've been using this metric to measure how much of a impact every one of the different diseases causes worldwide. And they've been doing this since 1990. And every single year since 1990, pneumonia is number 1 or number 2 greatest burden of disease worldwide. It's the most common cause of death due to infection in the United States and worldwide. It causes 1/4 of all the deaths due to lung disease. The majority of cases of acute respiratory distress syndrome, even before COVID-19, the majority of ARDS cases were from pneumonia, and it's the most common cause of sepsis. And sepsis and ARDS are the 2 most important critical care illnesses at present. And so really, if you care about critical care medicine at all, you have to understand that pneumonia is a big deal. In fact, we've built our ICUs in this country to be size enough to handle pneumonia. And that's what dictates the size of the ICUs we have. And the reason COVID-19 is such a problem is because it's overwhelming the capacity of these ICUs that we have. So what is pneumonia? I think we all need to take a minute to think about what the disease is that we're talking about. And in fact, it's an unfortunately difficult situation because it doesn't have a clean and rigorous definition. Other diseases and syndromes like sepsis or like ARDS have adjudicating bodies who meet every few years and agree on very rigid definitions of what those are. And that means everybody who's studying it and every clinician who's treating it sees it the same way. Everything that is ARDS is called ARDS and everything that's not is called not. For pneumonia, it's up to the physicians and the clinicians to decide for themselves if they think it's a pneumonia or not. And they all think that they know it when they see it, and they don't all agree on what it is. The general principles are shown here in red. There's an exudate or a consolidation in the lungs, a filling up of the lungs with fluid, and that's the defining feature of pneumonia. That's detected in 2 different ways. One is with the stethoscope, so it's called auscultation, and the lungs sound different when there's liquid mixing in with the air. And then it's also most commonly identified in imaging using chest x-rays or CAT scans. And in both the CT and the chest x-ray, what should be open air spaces, which are black on these imaging modalities, the fluid-filled spaces show up as white, and that's pneumonic regions. So this is pneumonia, liquid filling up what should be air spaces in the lungs. The other defining feature is that it's caused by an infection. And that also is really a bit of a namby-pamby definition, because we more often than not don't know what the microbe is that's causing the infection. That infection is presumed or deduced more often than actually identified. What are the microbes that cause infections? Those are highlighted here in this slide. And if you take a moment to look around, you'll see that, again, most cases go with a microbe unidentified, but when a microbe is identified, it can be very often a virus or very often a bacteria, and a whole lot of different viruses and a whole lot of different bacteria. And there's, frankly, for those of you who are microbiologists or interested in these sorts of things, there's nothing in common amongst these viruses or these bacteria. There's not some microbiological characteristic that means this is going to be good at causing pneumonia. In fact, the only thing that's really common about these things is that they're very common. They're ubiquitous. We see these things all the time. We don't get pneumonia because we ran into a pneumococcus or one of these other microbes. We see these microbes all the time. The vast majority of healthy toddlers have pneumococcus in their nose. 20% of the parents of toddlers have a pneumococcus in their nose, but they don't have pneumonia. Pneumococcus it's responsible for pneumonia, and it's the most common bacterial cause of pneumonia. The most common viral causes were rhinovirus, influenza, human metapneumovirus, respiratory syncytial virus, until this year, when now SARS-CoV-2 is a new virus that's causing lots of pneumonias. But very much like what I'm trying to communicate about these others, it's pretty wimpy. And I know that sounds a little bit heretical because it causes so much fear and it's such a big deal. But if you look at the statistics, a large fraction of people don't end up feeling any symptoms at all when they're infected with SARS-CoV-2. the majority of people infected with SARS-CoV-2 have a mild disease that would not require any hospitalization. Another smaller fraction require hospitalization with moderate disease and ICU admission if they have severe disease, and the very small fraction die. So this is SARS-CoV-2 is, again, it's a very, very common, 11 million cases in the US so far, very common infection, but it is more often than not, it is not a severe disease. It's kind of wimpy. So this is the general truism of the things that cause pneumonia. So why do they cause pneumonia? What's different? Well, I think before we understand that, we have to think a little bit about the respiratory tract and what we know about how the lungs work, the structure and function of the respiratory tract. The respiratory tract really can be broken into two segments. There's the airways, which are really just pipes for moving air in and out, and the alveoli, which are these blind sacs at the ends of those pipes, which is where the air comes down into and where gas exchange occurs. So you've got airways for moving air and then alveoli where gas exchange occurs. The airways begin in the upper airways, in the nose and the mouth, and there's just complete openings to the outside world. All this outside air comes in through the upper airways, through the larynx and into the trachea, which is the large tube leading down into our chest, which then splits into two different tubes, the bronchi, which go left and right into the right and left lungs. And then the bronchus will split and branch and branch and branch and branch, every about 23 or so branches in a human lung until you get to these very smallest branches and then these little blind sacs at the end. And those are called alveoli. And air is just being piped down here to the alveoli, which is the business end of the lungs. And in the alveoli is where gas exchange occurs. That's important because when the air gets down to the alveoli, it meets the blood. This is a pulmonary artery. This is coming from the heart. It contains deoxygenated blood. All the blood comes back to the heart through the veins, gets pumped to the heart, to the lungs through the pulmonary artery, comes down along the airways, through the arterioles into these pulmonary capillaries which surround the alveoli. That's where gas exchange occurs. The deoxygenated blood goes from blue to red when it becomes oxygenated, and then returns from the pulmonary vein to the heart, where it can then be pumped to the rest of the body now. And this is why we have lungs, so that we can oxygenate our blood. This is the big picture, the macrostructure of the lungs. Here's the ultrastructure, or the microscopic view of the lungs. Again, the airways, big airways, smaller airways, and then the alveolus at the very end, these blind sacs. You have a lot of structure around these larger airways, big columnar epithelial cells, glands, smooth muscle cells, lots of connective tissue, collagen, cartilage. You have lots of different structure that's supporting these large airways, but it gets smaller and smaller as you move into the smaller airways until you get to the alveoli, which are incredibly thin. This is air, and this is air, and this is a very thin alveolar septus, a single epithelial cell on top of a single basement membrane, and then this blood vessel here, very thin. This is a cartoon, but this is an actual transmission electron micrograph of the alveolar septus. And these are pulmonary capillaries here. And these are red blood cells. All of these dark blobs are red blood cells. So they're going to pick up the oxygen. This is airspace. This is airspace. And the oxygen needs to move from this alveolar airspace across that little barrier into this red blood cell, so it can be returned to the heart and pumped to the rest of the body. So that's the alveolar structure. It's incredibly lacy, and it requires this very, very thin barrier. And how thin is that barrier? Well, it's extraordinarily thin. I mean, it's 0.5 microns, which will mean something to somebody out there in the audience, but not much to most of you. So I try to provide some perspective on what 0.5 microns means. A human hair, as shown here, one single human hair, the thickness of that hair from side to side is about 50 to 100 microns. So this distance between the inhaled air and the red blood cell in the pulmonary capillary, that's on the order of 1/100th the thickness of a human hair. So it's an incredibly thin barrier. It's the thinnest barrier of our body by far, the thinnest barrier between the outside world and the inside world. And that thin barrier is not small, it's huge. This is a 2 micron bar to give you some scale down here. This is 100 micron bars, 100 microns, so this is a 50-fold lower magnification, and then down here is 500 micron bar, so another 5-fold magnification. You can see these are the surfaces of these alveoli. So this is a scanning electron micrograph instead of a transmission electron micrograph. And you can see the surfaces really nicely. And as we zoom out, this is the smallest airways, the bronchioles that then form these alveolar ducts, and then these are cross sections through these alveoli. These are what's highlighted here. But this surface area is huge, it's vast. If you were to unfold the alveoli and all the surface area of an individual, it's the surface of a squash court. So it's very large. Each of these 2 players on the squash court has an entire surface that size folded up inside of their chest cavity. So that really large, really delicate, thin tissue has to be protected. Well, maybe we can just keep the air out, but obviously we can't do that. We have to breathe. We have to breathe all the time. It'd be convenient if you wanted to protect yourself against SARS-CoV-2 to say, I'm going to go to the grocery store. I just won't breathe for the next 20 to 30 minutes. But you can't do that. We have to breathe all the time. We have to take several breaths of air. We take usually a breath every 4 seconds or so, half a liter per breath. It adds up to about 11,000 liters of air every day into our lungs. We have to pass that much outside air through our lungs, through this delicate, fragile, lacy environment. And that air is contaminated. It's filled with microbes. As we all know now, SARS-CoV-2 is transmitted through the air. It's aerosolized. Lots of viruses and lots of bacteria are in the air. There's also fungi and particulate pollution and pollens and all sorts of things in the air. All of those have risks of getting down there into those alveoli and disrupting that alveolar function. And that would be disastrous. So how do we protect ourselves from that? There's multiple good ways. Most of us aren't suffering from lung diseases, and because our lungs work really well. First of all, our lungs branch beautifully, and that's shown here. As you can see, that leads to when things are coming in with inertia, boom, they're going to impact on the side of an airway. Even if it gets past there, boom, it'll impact here. When it gets to here, if it's lucky enough to get there, boom, it'll impact here. It's very unlikely to get all the way down into the alveoli. And if it hits the side of these airways, then there's a beautiful system for getting it up and out of the lungs. I told you the columnar epithelial cells included multiple types of cells, but they can be broken in general into 2 types. Secretory cells, which basically produce liquid and mucus that lines the surface of these airways. This is the air up top. These are the cells, and between them is some liquid and a mucus layer. And then ciliated cells. These ciliated cells have cilia that stick out of the top, and they sweep all the time. They're sweeping in synchrony and moving this mucus blanket forward. So if they're moving from the smallest airways up through the trachea to the epiglottis, where it's then swallowed, and all the things that impact on the sides of those airways end up getting eliminated in our stool through our GI tract. So it's a wonderful system for eliminating things from stopping them from getting to the alveoli. But it's not perfect. Things will get through. There aren't any valves or doors, so things will sometimes land down there in the alveoli. This is a scanning electron micrograph of the alveolus. These are type 1 epithelial cells. There's a type 2 epithelial cell. Everything is an epithelial cell except for this. This is a different cell. It's an alveolar macrophage. Alveolar macrophages live on the surfaces of the alveoli, and they just crawl around down there. They move between these pores of cone, the holes between alveoli, and they are just patrolling and looking for trouble. So if something does get through all of these great defenses and land down here, the alveolar macrophage will find it and eat it and either destroy it or move on to this mucocilia escalator to get swept out. So it's a wonderful system. It should work. It should protect us. But if there's too many microbes, or if the microbes are too virulent, then it's not enough. Those defenses are overwhelmed. And when that happens, that's when we need an immune response. And that's basically calling in the troops, bringing in reinforcements to fight against these microbes. That's shown here. This is an alveolus. Now, instead of one single alveolar macrophage, you can see there's a whole bunch of cells in there, and there's this gross fiber meshwork there. In cross-section through a different sort of a microscopy, you can see that this alveolus is filled with fluid, this liquid, and with these cells, these are immune cells. This liquid, this is great. This is a fantastic way to fight infections. The liquid contains antibodies and other antimicrobial factors which can kill microbes, and these leukocytes are really good at killing microbes. But you can also imagine this is horrible for the lungs. This alveolus is not going to be good for gas exchange, but it's a price we're willing to pay in order to get those microbes out of there. So what are these cells? These are called neutrophils. That's spelled out here. Neutrophils are professional immune cells, and they're the first ones on the scene when there's trouble in the lungs or elsewhere. Neutrophils are defense cells. These are microbes in this cartoon within an alveolus, and these microbes are being ingested by a process called phagocytosis, which is a cellular process of eating. That's Greek for eating, phago. And these neutrophils are eating these bacteria. This is a cartoon, but here's it is happening in real life. This is a neutrophil. This is a bacteria called Pseudomonas aeruginosa. And there's an arm coming around this way. There's an arm coming around this way. They're just about to pinch off. This bacteria is getting enclosed, sequestered away from the rest of the environment. Here, this is a phagosome that's more intact, and you can see this is a bacteria. Now it's coming in and out of the screen instead of caught sideways. Same sort of healthy looking bacteria in cross section, but look at this one up here. This one is now swelling and becoming hypodense. This is on the way to being killed by this neutrophil. And here, it's already been popped, it's being degraded. This is what neutrophils do. They ingest and kill microbes. They're really good at it. That's one of the ways. They also can extrude these meshworks, which can have antimicrobial things to kill extracellular bacteria. They can signal to other immune cells, but also really importantly, the things that they use to kill these microbes, well, those are dangerous. They use things like proteases, which are, like meat tenderizers. This is how you chop up things. They have proteases that they use to kill bacteria. Those can also damage the lungs. And they generate reactive oxygen species. And in particular, neutrophils are really specialized for producing a reactive oxygen species called hypochlorite. And hypochlorite is the active ingredient of Clorox bleach. So these cells are making Clorox bleach in your lungs, and that is dangerous, as you can imagine. So these processes have to be really carefully regulated. And one way we regulate neutrophils is we keep them out of the tissues. We keep them in the blood most of the time. So here's a alveolar sepsis again. This is a capillary. Here's a neutrophil that would normally be in the capillary lumen, the inside of the blood vessel. But because it was an infected lung, what happens is the neutrophil crawls out. It moves between two endothelial cells, crawls between the connective tissue in contact with another cell till it gets into the airspace, crawls between two epithelial cells and into the air spaces. And this process of neutrophil recruitment was a focus for us for many years. We learned many things, and they're summarized down here in this review about how neutrophils get into the lungs. And it's a beautiful and elegant and complicated process. For one thing, neutrophils are always there. They're marginated in the capillaries. They're not in the air spaces, but they're inside the capillaries. And that's because the vasculature of the lung is a low pressure system, and the diameter of the pulmonary capillaries is smaller than the diameter of a neutrophil. So it's just squeezed and deformed to fit through. So it concentrates neutrophils in the pulmonary capillaries. They're there and ready at the beck and call if needed. When they migrate out of the capillaries instead of the postcapillary venules, which is where neutrophil recruitment occurs in most of the other non-pulmonary tissues. And they use adhesion molecules to interact with other cells and with the matrix that they have to find and recognize. And a lot of the adhesion molecule rules are described in this paper, and I won't go through those in detail, but I want to illustrate two basic principles, which are that lungs are different than other tissues. So here's an example of CD18, or beta-2 integrin, is an adhesion molecule that was first really became prominently recognized because it's the adhesion molecule underlying leukocyte adhesion deficiency type 1, which is a disease that humans and cows and dogs get. And if the people lack this CD18, or if animals are engineered to lack the CD18, neutrophils cannot get out into most of the tissues. They simply won't show up there. But in the lungs, they can. But it's not as simple as they don't need it in the lungs, because if you use more sophisticated systems to really try to understand when they're needed and when they're not, in some cases, for some stimuli, like for example, a Pseudomonas aeruginosa, you do see that there is a role for CD18 for neutrophil recruitment, whereas for other stimuli, absolutely no reason whatsoever, no requirement at all. So it's a very elegant, very complicated process. And we spent years trying to understand the rules that dictated how and when and why neutrophils got into the lungs. And that led us to focus on transcription factors, because we realized that the adhesion molecules needed to get turned on in the lungs so that the neutrophils could recognize them and crawl in the right places. That colony stimulating factors needed to get turned on to stimulate neutrophils to be produced in the bone marrow so that they'd be available to be recruited to the lungs. And chemokines need to be expressed. Chemokines are basically come hither signals that are expressed in the lungs and to direct neutrophils to come in those directions. All of these, the genes for all of these things the genes are in all of our cells, but they're not being expressed unless they're needed, unless there's infection or damage. And they're regulated by things called transcription factors. And one that we found is really important is NF-kappa B rel A, and that's highlighted here in green. These are lung cells, and you can see the nucleus are these dark holes, and they're dark because there's no NF-kappa B rel A in those nuclei. But within minutes of being stimulated, all of a sudden rel A is, these are stimulated by bacteria. These transcription factor NF-kappa B rel A is moving into the nucleus. The nucleus is very bright, and this transcription factor is binding to DNA, turning on the expression of these things, chemokines, colony stimulating factors, and adhesion molecules. So now these cells are telling the neutrophils, this is where you need to be. You got to come out. We need you here. So this was a focus for quite a few years of us. After focusing on neutrophil recruitment, we've started focusing on the transcriptional regulation of immunity in the lungs. And that led us to these general concepts. One is that we need multiple different types of gene programs that are regulated by multiple different types of transcription factors. One major type of gene program is called immune resistance, and immune resistance is really basically the weapons we use to kill microbes. And it's sort of the equivalent of a sword. And the immune resistance is things like calling neutrophils into the lungs to fight microbes. And that is dictated by multiple different transcription factors, which we highlighted in this review with smaller circles, but the big circle, NF-kappa B, was a really important one. And for example, if you lack NF-kappa B rel A, you can see some tissues. This is skin. This is the surface of the skin. This is skeletal muscle underneath here. This is a lake of bacteria. And for those of you who know how to look at histology, there's no immune response around here. Absolutely none. In the absence of NF-kappa B rel A, you cannot defend yourself against these bacteria. These are spontaneous infections that are happening with just because that transcription factor is missing. In the lungs, things were a little different. There were lakes of bacteria again, but these alveoli were now filled with immune cells. So again, the lungs are different than other tissues. Immune cells could get in, but they could not mount an effective defense without NF-kappa B rel A on board. But it's dangerous. We hear a lot about cytokine storm. You hear about it with SARS-CoV-2. You've heard about it with avian influenza. You've heard about lots of different settings. It's an important concept, and it applies in some cases. Cytokine storm is really an excess amount of these sorts of transcriptional activities, and it leads to too much of these activities lead to things like acute respiratory distress syndrome, shock, multi-organ failure. These sorts of things result from too much, but too little means you're going to get an infection. So these dangerous things have to be balanced by other sorts of programs. And we found that there are multiple transcription factors that influence those. And a really important one was one called STAT3. And STAT3 is important for driving this tissue resilience. So instead of attacking microbes, tissue resilience is really more like the shield. You want both a sword and a shield. And this is the shield, is tissue resilience. It helps protect you from the onslaughts of the microbes and from the onslaughts of your own dangerous immune activities. In the absence of STAT3, we can get rid of bacteria or viruses pretty well. But the number of organisms don't differ between these two lungs that do or do not have STAT3, but these lungs that are leaking STAT3 are filling up with fluid, as you can see here and as is measured here. The absence of STAT3 really meant that you had too little tissue resilience, so you had too much damage from these immune resistance mechanisms. So just like NF-kappa B has to be modulated, these also have to be modulated. Too little of these things will lead to ARDS, shock, or multi-organ failure. But too much of these tissue resilience pathways will lead to infections, but they also can lead to dysfunctions of the tissues, including chronic long-term diseases. So this a really tight balance is what's absolutely critical here. Well, immune resistance and tissue resilience aren't simply static factors that all of us have and that have the same the rest of our lives. They change between us. All of us have different levels of immune resistance and tissue resilience. And for all of us, we have different amounts of tissue resilience and immune resistance throughout our lifespans, influenced by things like age, as highlighted here, and by exposures like cigarette smoking or alcohol abuse or other pollutants and other things. Tissue resilience usually starts very high and decreases over the lifespan. And immune resistance is even more dynamic. It usually starts very low. We're born with very, very low immune resistance. But within our first years of life, immune resistance increases dramatically. And by late childhood and early adulthoods, we really have peak immune resistance. And then as we get older, that starts to wane again. And this early increase in immune resistance has been interesting us a lot recently, and I would like to share with the group for the next few slides is what's responsible for these early increases in immune resistance. And we think one of the major factors that's driving this increase in immune resistance, is the fact that what we need resistance against are these wimpy but ubiquitous pathogens that we see all the time. And one of the things is when we get over these, we're building resistance against those microbes every time we get over one of these infections. And a lot of that comes down to a type of immunity called heterotypic immunity, which involves recognition of what are called the heterotypic microbes. And heterotypic means sort of the same but different. Hetero is different and typic is just type, different type. And so I mentioned, for example, the easiest one for everybody to recognize is influenza. You know, you need to get an influenza vaccine every year because influenza is different. Every year it comes by. It's a new strain of influenza. We don't have homotypic immunity against next year's influenza strain. So we don't have any homotypic immunity, which would be neutralizing and would completely prevent an infection from that strain that's going to come next year. But we do have immunity against influenza viruses because we've seen influenzas. We've seen lots of influenzas. By the time I'm my age, I've probably had multiple infections with influenza viruses. So I have memory against influenza, just not the same influenza, but there are parts that are conserved and are similar. It's never as good as the homotypic immunity, but it is a whole lot better than the absence of any immunity. This heterotypic immunity in general, it's much better than having no adaptive immune responses, but not nearly as good as having homotypic immunity. I gave you influenza as an example, but we think this probably applies to all respiratory pathogens. That influenza is a very common viral cause of pneumonia. Pneumococcus is the most common bacterial cause, and these are pneumococcus data here. This was an input of bacteria, and you can see in a naive lung that these bacteria can grow to 10 to 100-fold in just a matter of 24 hours in a naive lung. It's a very severe infection, pneumococcus. But this is a serotype 3. Pneumococcus doesn't change seasonally, but pneumococcus just comes in a wide variety of flavors. There's over 100 different serotypes of pneumococcus. And so serotype 3 grows in the lungs, but if previously we've never seen a serotype 3, but we've seen other serotypes like 19F and 35B, we can have a profound improvement in lung defense against that serotype 3. Even though we never saw serotype 3, we, instead of having 10 to 100-fold increase, this is a log scale, we'll have a 100-fold decrease. So 1% of the input, 99% eliminated in a matter of 24 hours. So it's a really profoundly protective immunity, heterotypic immunity. And one of the things that I think is most exciting about heterotypic immunity and really sets it apart from homotypic immunity, we think that heterotypic immunity really resides in the lungs. We're learning more and more these days that the lungs have their own immune system that's really very different. It's reliant on, but very different from the systemic immune system that's involved in all the other tissues, including lymph nodes and spleen and blood. In a naive setting, naive lungs, the left lobe or the right lobe doesn't really make any difference. They have approximately equal crummy defense against a pneumococcus in the lungs. But pneumococcus typically causes a lobar pneumonia, so a single lobe will be infected and the other lobes are not. And if, for example, the left lobe was infected originally with a serotype 19F, that will establish heterotypic immunity against the serotype 3. And if that serotype 3 comes back into the left lobe, which had been previously infected, or into the right lobe, which had not been previously infected, you have profound differences in the bacterial clearance of those lungs. The ipsilateral lobe, the left lobe that had a previous experience, is able to very rapidly clear those microbes. But the contralateral lobe on the other side, it can't do it. And if you think about it, the blood has equal access to both sides. The circulating antibodies, the central memory lymphocytes, all of these systemic immunity can get to both sides equally well. Something is changing inside the lungs, and that's really driving a lot of our thought these days. What is it that's in the lungs and changing? And it's a lot. I'm only going to show you a couple pieces of it today, but there's a lot of things changing in the lungs. This is an experienced lung. You've seen several lung slides now, so hopefully you're starting to get the idea. This is a conducting airway, alveolar ducts. These are a bunch of these lacy alveoli. cut through in section. And now if you're getting to be developing a bit of an eagle eye, you might pick out there's some blue dots down here that I hadn't really focused on before and read your attention to, but now I'm trying to call those out. And if we blow this up and look, what are the blue dots? Now we're going to stain them with different sorts of colors to highlight what they are. And if they're aqua, that means they're CD4 T cells. And CD4 T cells are something all of you probably know about from AIDS. HIV attacks CD4 T cells and destroys CD4 T cells. And that's why HIV was such a problematic disease and really made people so immunosuppressed, because CD4 T cells are so critical to the immune system. Well, some of these CD4 T cells seem to live in experienced lungs, these aqua cells here. And then B cells are highlighted in red, and there's red B cells in these areas as well. And B cells are important because B cells make antibodies. And all of you know about antibodies. There's been a lot of talk about antibodies with SARS-CoV-2 and whether we do have antibodies and if vaccines can generate antibodies. The cells that make antibodies are B cells, and there are B cells in the lungs. And so that's led us to ask, what are these T cells and B cells doing in these lungs that might be protective? Well, we and many others have come to recognize there's this group of lymphocytes, including T cells and B cells, that are called resident memory lymphocytes. And these are cells that live in the lungs. They don't recirculate, they don't go back into the blood, they don't go back through lymph nodes, they stay in the lungs. And they really have different behaviors. And one thing is they're much, much faster in response now. So if a lung is infected that's never seen a microbe of that kind before, the very first coronavirus it's ever seen, the very first pneumococcus it's ever seen, it will take on the order of 6 to 10 days before you can get T cells to be really active in those lungs. For something like a pneumococcus that doubles every 20 minutes, 6 to 10 days is a lifetime. That's much too slow. We can't have that. If you have resident memory cells in the lungs, these are heterotypic. They recognize that they've never seen this pneumococcus before, but they've seen another one. Then they can respond very fast. Within a day, they're all fully revved up and acting in those lungs. And not only acting, they're acting a little bit differently, and they're acting a little bit better than these lungs from a naive lung. So they're faster and they're better if they're resident memory cells. But what do they do that helps? And we've really been recently outlining that. What we think one of the most important things that the T cells do there in the lungs really relies on what I already told you before about getting those neutrophils into the lungs. For a general principle that's really important for lung defense that we're learning is speed matters. The rapidity with which you can get those immune responses to occur, to recognize the microbes and to start killing those microbes is a major determinant of what's going to happen for the rest of that infection. And we typically thought of adaptive immunity, which is where T cells and B cells are, as being very slow, much too slow. We needed innate immunity, which was fast, like neutrophils. What we found recently is that adaptive immunity actually hurries innate immunity. It accelerates the process of the immune response. And you can see that here. This is 24 hours after infection. This is experienced animals that have seen a different pneumococcus before. There's no difference in the naive or experienced lungs by 24 hours. Neutrophils are going to get there eventually. But in those early time points, 7 hours after infection, the experienced lungs have a lot of neutrophils coming in before the naive lungs do. And what's doing that? Is it the T cells, and what are they doing to do it? Well, we looked at that by asking, what are the come hither signals that are present? And there's multiple, and we measured them. And one in particular stood out. It's a chemokine called CXCL5. It was much higher in the experienced lungs than in the naive lungs. And so we blocked that particular signal. And if we blocked that signal, the neutrophil recruitment was decreased. So it really said this increase was essential. And we know this increase comes from epithelial cells. It's not coming from the T cells themselves, but we learned in this paper that's highlighted down here that the T cells are instructing the epithelial cells to hurry up and make this CXCL5 better and faster. And is this CXCL5 and neutrophil recruitment really dependent on the T cells? And the answer is yes. Here we devised a strategy that we could eliminate the CD4 T cells from the lungs using this agent called GK1.5 and the way that we administered it. And when we did that, it was very effective at getting rid of those T cells. And that led to less of that come hither signal and less neutrophils at that early time point. So the T cells, really what they're doing is they're accelerating our response, our recognition, and our ability to get over those microbes that land down there in the lungs. What about B cells? B cells is a much newer area of research when it comes to resident memory lymphocytes. They were first published in 2019, so we're all, as a community, still learning. But these are some of the data that we have related to resident memory B cells in infected lungs with pneumococcus. There are antibodies that show up in the air spaces of the lungs during a pneumococcal pneumonia, and these antibodies are heterotypic, so they recognize different serotypes. And these antibodies that are in the air spaces of the lungs are different than the air spaces that are in the blood. If we take the B cells out of some lungs and we stimulate them in test tubes, they start to make antibodies, and these antibodies that they make are heterotypic. They recognize different flavors of the microbes. And so therefore they're capable of making these things. And if we take the B cells out of the lungs, now these lungs are more susceptible to a heterotypic infection. So these B cells really have essential roles in generating antibodies locally in the lungs, and that helps to protect these lungs against this heterotypic microbe, in this case, a heterotypic pneumococcus. So I like cartoons, I like drawings, and this is a lot easier to communicate, I think, than data, especially with this wide audience. I appreciate you coming on this journey of immunology with me, especially for some of you who this sounds like alphabet soup. But the idea is we're born with these naive lungs. These don't have any cells that recognize microbes yet. Specifically, they don't have any adaptive immune cells. But we live in a dirty world that's filled with microbes, and all of us very soon after birth start to get respiratory infections. And these respiratory infections are common. And they're things like rhinovirus and influenza and now SARS-CoV-2. And they're things like pneumococcus and Staph aureus and Haemophilus. And when they land in the lungs, they change the lungs. And they change them in ways that I've highlighted that include T cells and B cells, resident memory cells. And they also include other lymphocytes, which I didn't get a chance to talk about today. And also you can see that some cells are changing color now. And that really emphasizes that even the lung cells themselves behave differently now because of these prior exposures. There's a really different new memory of prior exposures that occurs in the lungs. And we're really working hard to try to understand what that memory is now. And it's important, we think, especially for recognizing heterotypic infections, infections that are similar to, but not identical to what we've seen previously. Well, does that apply to COVID? I mean, could it? Because COVID is new, right? We haven't seen it before. But sharp-eyed individuals might have noticed that coronaviruses were already a cause of pneumonia, the 7th most common cause of pneumonia hospitalizations in the United States already before COVID-19 occurred. And that's because there are 4 endemic human coronaviruses that are circulating among us all the time. They peak every winter. They're like these others I'm talking about most of the time, very wimpy, but occasionally very severe causes of pneumonia. They usually cause common colds, and they're very often these days to be contrasted with SARS-CoV-2 called the common cold coronaviruses. But they're endemic and they're capable of causing pneumonias. They're sufficiently likely to cause, here's their names, which are not very interesting names. They're sufficiently likely to cause pneumonia that they've been included in panels, diagnostic panels. Just like you get a SARS test by swabbing the nose, you can swab the nose and do a PCR test instead of for SARS-CoV-2 for other viruses. And there's a respiratory pathogen panel that we use at our hospital at BU, the Boston Medical Center, that measures all 4 of these in addition to a couple dozen other microbes, microbes, both viruses and bacteria. And so we asked, have we seen patients who have had these coronaviruses before? And if we have seen patients with those coronaviruses, do they have different outcomes with COVID-19? And so here's basically the setup of the study. We looked over the last 5 years up until March 11th, and we picked that date because we had our first COVID case at BMC on March 12th. And we looked for all the patients who had been swabbed and tested for respiratory pathogens and asked, were those patients who were swabbed in the last 5 years, were they negative for the coronaviruses or were they positive? And a small fraction of them were in fact positive. We had about 16,000 subjects who were swabbed, and about nearly 1,000 of them turned out to be positive for coronaviruses. We then asked, did anybody show up March 12th or later and ask for a COVID test because they had influenza-like illnesses? And it turns out that about almost 2,000 did. Most of them didn't, obviously. This is a very short period we looked at, about 3 months, March 12th to June 12th. But about almost 2,000 people did show up, and most of them had been people who were coronavirus negative previously, but a group was previously coronavirus positive. And we asked, did this group differ when they were tested for SARS-CoV-2 from the other group? And that was really what we wanted to know. Was there different rates of infection? And if they were infected, did they have different outcomes? The table here on the right is for the aficionados in the group. Basically, it shows these groups, whether they had a history of coronavirus or they didn't, are pretty similar. They're not identical, but they're pretty similar. So here are some of the outcomes we saw. Who came in to ask for a test? Well, it turns out about 11% of the people who had been coronavirus negative came in and asked for a test, and about 15%, a little bit more, statistically significantly more, came back to ask for a test if they had been coronavirus positive. So that suggests that maybe people who had gotten a coronavirus are really maybe almost the canaries in the coal mine, the most susceptible to getting infection and most likely to come in and need some more influenza-like illness testing after March 12th. So a slightly larger fraction. But if you looked at who got SARS-CoV-2 or not, the fraction was the same. About 25% were SARS-CoV-2 positive in each of those two groups. So this suggests that a prior experience with endemic coronaviruses really is not likely to prevent a SARS-CoV-2 infection. Well, what was the outcome? A lot of them were hospitalized, and that's because if you remember back in March and April and May, SARS-CoV-2 tests were very hard to come by, and only the sickest patients were getting them. And over half of the patients were hospitalized who got these SARS-CoV-2 tests back in those days, and there was no difference between groups. So it didn't prevent hospitalization. But what about moving further into the category of from moderate into severe disease, where you have an ICU admission? That's where we really saw some interesting data. About 28% of the SARS-CoV-2 positive group that had been hospitalized with a no endemic coronavirus history ended up in the ICU, but less than 5% if they were patients who had been previously identified with a coronavirus. Statistically significant difference, even after adjusting for all of the potential confounders we could identify. So it really suggests those who recovered recently from an endemic coronavirus might be less likely to end up in the ICU during COVID-19. And if we looked at what happened during that hospitalization, did they die or not during the time that we monitored them? You can see that those who did not have a history of coronavirus, about 20% of the hospitalized patients ended up dying over that time frame. But those who did have a history of endemic coronaviruses, only again about 4% of them ended up dying during that time frame. Statistically significant difference, again, really suggesting that a history of recent prior coronavirus infections may make you more likely to survive COVID-19. So that suggests to us that heterotypic immunity probably really does impact COVID-19 and SARS-CoV-2. And that's an area of future research that we're really emphasizing. I think a lot of what we've learned about pneumonia over the years applies to SARS-CoV-2. And a lot of the questions that are important for pneumonia are now really important for addressing COVID-19 and SARS-CoV-2. And this cartoon comes from a workshop that we put together. Charles Delacruz, Rich Wunderink, and I bring together 29 pneumonia researchers from around the country down to the NIH, and we could all spend 2 days together locked in a room to try to decide what is the most important pressing questions related to pneumonia. And there's tables full of pressing important questions for pneumonia. I recommend this paper to you if you're interested in saying, I want to study COVID-19, because I think if you replace the word pneumonia with COVID-19, this paper is basically marching orders for what needs to be done for COVID-19. And none of this below the line here was in the paper, but sort of paraphrasing what you might say to apply this to the current situation we're in. Questions include, how do age, exposures, comorbidities alter immune defenses against SARS-CoV-2? How do we understand the susceptibility that happens before you get that infection? Can we measure an individual's susceptibility to SARS-CoV-2, and can we counter that susceptibility? Second, during the pneumonia, what determines whether somebody recovers quickly, declines quickly and dies, or has a long and prolonged and difficult course? We need to understand the factors that make this COVID-19 mild or severe by constraining the virus or preventing the injury caused by the virus and the immunity. And we need to be able to measure and manipulate those specific pathways in specific individuals so we can use precision medicine approaches to tailor the therapies appropriately. And then finally, host consequences was already clear before COVID-19. After you survive pneumonia, there are extrapulmonary consequences and long-term consequences, cognitively, emotionally, cardiovascularly, pulmonary. Lots of systems are compromised. And now this concept of long COVID and COVID long haulers is really bringing this to bear. And we need to understand the biology of that. And I want to emphasize these questions will now be important for COVID, but I also want to emphasize that all the discoveries being made about COVID right now come back and apply to pneumonia as well. And that's what really gives me hope, what really makes me excited right now. One thing that really gives me hope is the fantastic news we all have learned recently about vaccines against SARS-CoV-2 in the last week or so. 2 already look very promising. There's more on the way. I think that will be game-changing for SARS-CoV-2 specifically. But the discoveries that are being made about SARS-CoV-2 will apply to pneumonia. And so many people now are becoming pneumonia researchers thanks to COVID-19. I spent years of my life trying to convince more people to study pneumonia, ending essay after essay with statements like this: whatever the reasons, means that an important cause of human suffering is relatively underserved. And by underserved, I meant we could not get enough researchers to come and put their great minds on this problem. Well, that's ended. This is PubMed just searching for the word pneumonia. And just the publications on pneumonia has boomed in the last year. The discoveries being made are mind-boggling. And this doesn't, if I search COVID, it's a much higher number. This is just, I searched pneumonia, and the numbers of investigators bringing their expertise to bear on this problem is going to mean not just improvements for COVID-19, but I think for all of pneumonia, which means all of ARDS, which means all of sepsis, which means all of critical care. I think things are going to be very hard for the next couple months, really hard for all of us, harder than we've experienced so far. But I think things are going to get better. They're going to get better very soon for COVID. Within a year from now, we'll be in a much, much better place a year from now. And I think they're going to be much better for all of critical care medicine within 5 to 10 years because of this huge scientific focus on pneumonia. I have 3 quick acknowledgement slides to finish. First, this is the Boston University lecture, and I want to emphasize how much I love being a Boston University faculty member. And this is why this slide tells the story. These are all faculty members who, while they were at BU and I've been at BU, I've been here 12 years now, have been co-authors and co-investigators with me. This is outstanding. I love this. BU is a place that people love to work together. Everybody looks at each other and says, how can we help? What can we do together that would be better than any of us could do alone? And that ethos is tantamount everywhere at the university. I love that. Some of that is due to institutional support of institutions within the institution, and that includes things like the Pulmonary Center. The Pulmonary Center includes clinical researchers and basic science researchers who are coming together to study lung biology and pulmonary diseases, and they do it from a wide variety of disciplines: cell biology and molecular biology, immunology, developmental biology, regenerative medicine, epidemiology, health services research, and more. So many people coming together and truly working together to bring those disciplines together to do things to fight pulmonary diseases, including pneumonia, but not restricted to pneumonia. I also want to highlight the Evans Center from the Department of Medicine, and that's run by Katya Ravid. And the Evans Center is really specifically designed to try to do multidisciplinary work, both inside the School of Medicine and across the entire university. They try to bridge the schools, they try to bridge the departments, and you do that through things like called affinity research collaboratives. And I've benefited from that. I led a pneumonia ARC several years ago, and now there's a COVID-19 ARC that's run by Marcus Bosman and Mohsen Saeed and supported by the DOM. And those sorts of multidisciplinary initiatives really help to build this sort of ethos here. And then the NEDL, the National Emerging Infectious Diseases Lab, first supported by the NIH, now strongly supported also by Boston University, and really brings together so many people to bring everything they can to bear to focus on infectious diseases from all possible perspectives and all possible disciplines. Again, really truly a multidisciplinary enterprise, and I love that. Academia is special because of the relationship between mentors and mentees, I think. And I want to highlight a couple of those. My mentors in particular for science were Joe Brain, who was my mentor during my doctoral thesis research, and Claire Dorschuk, who was my mentor for my postdoctoral research. And these are both role models for me. They had a profound impact on me. I try to emulate them in every way, including how I mentor. And these are some of my past mentors who have moved on, and they've all been incredibly important to me in many ways, but in one, because I want to emphasize that the mentor-mentee relationship is really two-way. And I've learned tremendously from every one of my mentees, including all of those here shown here. Some of them are now still even BU faculty, so they're colleagues. So I learn from them in very real ways on a day-to-day basis continuously. But others, always, all of them have stayed in science. They're all at academic research academic institutions or at science companies, biotechs or pharmaceuticals. And all of them made a profound impact on me. They changed my research, they changed the way I'm thinking, and they were a great benefit to my life. And this is the current team. This is who we are right now. Shown at left, these are all trainees that are in the what we call the pneumonia biology group. And those that are highlighted in yellow are trainees that I'm the primary mentor for. And on the right are faculty collaborators. And these faculty collaborators are all listed here because they're co-investigators on either current or pending research projects. And a lot of them involve COVID-19 and all of them involve pneumonia. All of this is a great interaction. I love working with these people and doing this pneumonia research. I thank you for your time to sharing today. I really enjoyed this opportunity to share my research with you. I hope it was of some interest to you, and I'm very happy now to stop and to take some questions if there are any.
Well, thank you so much, Jay. What a tremendous presentation. And now we'll take questions. The questions will be read, and I'll let you respond as they come in.
Are there many strains of SARS-CoV-2?
That's an interesting question. I don't think there are as yet many strains of SARS-CoV-2. SARS-CoV-2 is changing and evolving, so there are new versions of SARS-CoV-2 that have mutations versus other SARS-CoV-2 viruses that don't have those same mutations. And some of them are becoming more widespread, but they haven't really been identified as strains yet of these new viruses, at least not to my knowledge. So over time, I wouldn't be surprised if they might develop into multiple strains, but at present, there really seem to be largely one strain with some variation within that strain.
We all have resident memory T cells in the lungs, but only some COVID-19 patients develop severe pneumonia. Would you say that some of the differences among patients depends on their lifetime exposures in the lung that create a highly personalized profile of resident memory cells?
Yeah, that's an excellent question. And I would desperately want to know the answer to that question. So I can tell you what I think, but I can't tell you the answer to that question because nobody knows the answer to that question yet. And it's a really important question. We have 2 grants pending now to try to get the funding to address that question. I think, and 1 grant actually that we have received, which will begin to address that question. I suspect the answer is yes, based on the clinical data, which I shared with you today, which is that the heterotypic infections do particularly prevent the severe consequences, the lung consequences, the ICU admissions and severe lung disease in these patients without preventing the infections. The infections themselves seem to occur regardless of your heterotypic infection history. We think that probably heterotypic infection to coronaviruses is establishing resident memory lymphocytes, both T cells and B cells, against coronaviruses, including SARS-CoV-2, in the lungs. Nobody has demonstrated that yet, and there's no knowledge of that yet. There are heterotypic T cells and heterotypic B cells in the blood. So there are absolutely heterotypic T cells and heterotypic B cells that can recognize SARS-CoV-2 that were generated by other coronaviruses. Could have been earlier SARS virus, could have been MERS coronavirus, or could have been the endemic coronaviruses. The data does suggest that in particular T cells seem to be capable in the blood of crossing between the endemic coronaviruses and SARS-CoV-2. Not as much the B cells, but there's some evidence for that too. But for the lungs, there's no data yet, literally none, at least none published. And we're working hard on trying to answer that question. I think the answer will be that yes, there are resident memory heterotypic lymphocytes in the lungs, and variations in those resident memory lymphocytes in the lungs would be a very strong determinant of who gets a severe SARS-CoV-2 infection and who kind of laughs it off.
Do simpler life forms have such complicated immune systems?
Oh, that's an interesting question. And yes and no. So I talked about innate immunity and adaptive immunity. So things like neutrophils are part of the innate immune response, which is really inflammation. It recognizes stereotypical type of things that are common to lots of different types of microbes. So for example, it might recognize all gram-negative bacteria, but it won't recognize any gram-positive bacteria, and it won't differentiate two different gram-negative bacteria like a Pseudomonas aeruginosus and a E. coli. That's innate immunity. That is present in really even very, very, very primitive animals. It was really one of the major discoveries were what are called toll-like receptors, elements of that innate immune system. And one of the three people who got the Nobel Prize for that discovered the toll-like receptors in Drosophila, the fruit fly. So even very primitive organisms have innate immune systems. Plants have innate immune systems. But the adaptive immune systems, the T cells and the B cells, and that very specific immunity that differentiates just SARS-CoV-2 from an endemic coronavirus, or differentiates all coronaviruses from all the influenza viruses, those are in more evolutionarily advanced, I guess, organisms. So for example, all mammals have those, but fruit flies don't.
Will people that have had COVID-19 infections and recovered also need to have the vaccine? Will a different vaccine be needed every year?
Those are very good questions, and we all wish we really knew the answers to those. You know, so far, no vaccines are licensed, and the vaccines are only beginning to come out, and we're only still beginning to understand what a natural recovery from a SARS-CoV-2 infection means. It looks like the natural recovery from SARS-CoV-2 infection will be very similar to natural recovery from other sorts of respiratory infections in several senses. Patients who have moderate disease with SARS-CoV-2 and end up hospitalized with COVID-19, they so far seem to be having very good persistent memory, both T cells and CD4 T cells, CD8 T cells, B cells, and antibodies, all measured in the blood, mind you, not in the lungs yet, but all those measured in the blood, very good memory 6 months later. So if you were sick enough to get hospitalized, excellent memory 6 months later. And if we extrapolate from other coronaviruses, like this first SARS coronavirus, which caused problems in 2003, patients who survived the SARS coronaviruses in 2003, they still 17 years later have really good memory. So that would suggest that if you generate that really good memory against SARS-CoV-2, it will last. But back to your question about should you get a vaccine if you've already had an infection, I would say it's not as simple as if you got an infection, you're immune. Certainly, there are multiple cases in the US and in other countries of people getting over a SARS-CoV-2 infection and then getting a second one. That's a different one, sequenced, identified, and proved to be a different virus than the first one they had. So you can get reinfected. And in general, and I think this will apply to SARS-CoV-2, but the data aren't really there yet. But in general, the stronger the infection you have, the more severe an infection you have, the stronger and more lasting your immunity is. So if you have one of these milder SARS-CoV-19 cases, if you're diagnosed as SARS-CoV-2 positive and you self-isolate at home and you end up with a runny nose and headaches and fatigue, but you don't end up hospitalized, I would not count on that immunity protecting you. I would definitely want to go get that vaccine. And most importantly, you know, we'll get more data as the vaccines roll out. They have not rolled out into the community yet, and we'll have much better and more informed, clinically guided ideas of when and who should get the vaccine then.
Does your community have a sense of the factors that lead to long haul symptoms? What portion of patients have identified long haul symptoms?
Those are also excellent questions. For COVID-19, there's virtually no sense for what determines long haul symptoms. It's an incredibly important question. In fact, for pneumonia, it's not very well understood, period. But there's some information about it with pneumonia. And there are no simple answers yet, but now my impression from the community and from talking to many people who do this kind of research and from being pretty familiar with it is I think probably a general principle that's going to emerge is that the long haul symptoms emerge from acute injuries that happened during that acute infection. And I think some of the best studied are the cardiovascular sequelae. And an example of this would be studies done by Carlos Orihuela and who was then a postdoc, a graduate student in his lab and is now a postdoc in our group, Ankle Shenoy. They showed that during pneumococcal pneumonia, pneumococci were getting into the blood, which everybody knew, that's called bacteremia. But some of those got into the heart and actually into the heart muscle, and they could live in the heart, and they created little micro lesions, and they persisted, and they can cause little scars. That those scars might never really heal, and those scars, or even if they heal someday, they might persist for a long time and cause some cardiovascular anomalies, cardiac anomalies, and that there's good evidence for that with pneumococcus. Related to COVID-19, there was a very sobering publication, and related to the second part of your question, which is how many people suffer from long haul consequences. There was a very sobering publication in JAMA Cardiology about a month ago that brought back a bunch of, 100 exactly. They brought back 100 patients who had recovered from COVID-19, and they did cardiac imaging and cardiac physiology tests. And even for patients who weren't reporting symptoms, 80 of 100 had detectable cardiac anomalies after that, within 3 months after their COVID-19. So the statistics vary, with surveys. It's sort of 20 to 40% seems to report things like fatigue and chronic headaches and shortness of breath and things like that. But if you really look carefully using physiological and clinical tools, it might be higher and might be approaching 80% might have long haul consequences.
Do seasonal allergy episodes make someone more or less sensitive to lung infections? And this will be the last question.
Okay. That's a very good question too. And seasonal allergies. To be frank, I have to answer that question with I don't really know. There's been conflicting studies about allergic airways disease and asthma and how that intersects with COVID-19. And I think probably the preponderance evidence suggests that like other chronic respiratory diseases, asthma does increase your risk of getting and having severe episodes of COVID-19. But all of the data don't agree. And there are, these are still early days, and epidemiology is a difficult science. So I think that's not very well understood. And as far as seasonal allergies like rhinitis and, upper airway sorts of responses, I haven't seen any data, which doesn't mean it's not out there, but I'm afraid I don't know the data and I don't want to mislead anybody.
Well, Jay, I want to join all of the people virtually in thanking you for an incredibly stimulating talk in a very timely presentation about a subject that you're deeply involved in, but also is affecting everyone's life as it has before, but also today more than ever. So thank you very much, and congratulations on being the university lecturer this year. And we all look forward to applauding you when we're back in person. Thank you for being with us.
Thank you very much. Thank you all.